Hi, my name is Lane Hansen, and today we'll be looking into the DNA replication process. On the left, we see our DNA double helix, which is fed through the replication factory of proteins, separating the DNA into two template strands, which are copied and become half of a new double helix. Thus, the DNA replication process is semi-conservative. So let's look into this replication factory of proteins. Here we have our starting DNA double helix. First, the helicase unwinds the DNA into two single strands. The point where the DNA is separated into two single strands and where the new DNA will be synthesized is called the replication fork. Next, the SSB immediately coat the single strands of DNA to prevent reannealing. Then, the primase synthesizes short RNA primers needed to start the replication process. The new strands act as template strands for the new production of DNA, which is done by the DNA polymerase. The DNA polymerase displaces the SSB and then strings nucleotides together to form the DNA strand. The sliding clamp holds the DNA polymerase onto the DNA strand. Let's look deeper into this DNA polymerase. The two strands go from 5' to 3' in opposite directions. Polymerase enzyme can only synthesize in the 5 to 3 direction because it attaches the 5th phosphate group of the free nucleotides you see around to the 3rd hydroxyl group of the nucleotides already on the strand. The leading strand up here is continuously synthesized, while the lagging strand is synthesized in short pieces. These short stretches of DNA that make up the lagging strand are called Okazaki fragments. Since they are anti-parallel, the polymerase moves in opposite directions. Once the polymerase on the lagging strand hits the RNA primers, the RNase H is needed to remove that RNA primer so that the polymerase can continue the synthesis. The synthesis is continued, but a hole is made. That's where the ligase comes in. The ligase links the short stretches of DNA together to make the DNA continuous. And there you have it. Our DNA has been synthesized. Thank you.